Hello and welcome to the session of Biology from Ansh Academy. I am Mrs. Ranjana Dubey, who takes up biology for Ansh. I am MSc in Biochemistry from Banaras Hindu University. Ansh is an online based platform for providing education in NEET and JEE courses as well as foundation courses. Our director, E. Lazarus, and his family have been associated with the field of imparting education since 4 generations. Now, before I proceed with the topics that I have brought for you, I would like to talk about the NEET examination. Now, the NEET examination conducted by NTA has 90 questions in it from biology, 45 from the botany section, and 45 from the zoology section. Out of the 45 botany section questions from class 11th and 12th NCERT syllabus, there is just one lesson which every year gives 5 questions in the NEET examinations. Every year from the same lesson, at least 4 to 5 questions are asked and have been asked repeatedly ever since I was a NEET aspirant. It is still the favorite topics of the examiners. You guessed it right. There is a chapter which is so very important, and I want to tell you the name of that lesson. It is Principles of Inheritance and Variation. Now, before I start in explaining you the concepts related to this particular lesson, I have with me some questions of this lesson from the NEET examination. One by one, I would be taking you through those questions, and as we answer them, we would also be clearing our concepts in this particular chapter, Principles of Inheritance and Variation. So, I bring forward to you the first question in this chapter from the NEET, and that one is: which one from those given below is the period for Mendel's hybridization experiments? The options before you are 1856 to 1863, 1840 to 1850, 1857 to 1859, and 1870 to 1877. It is a very simple question, but only the student who really is thorough with the lesson will be able to provide with the right answer. And the right answer is option number 1, which is 1856 to 1863. Okay, now moving on to describing about the lesson and starting with Mr. Gregor Johann Mendel, who is the father of genetics. It was he, Mendel, who laid the foundation of the modern day genetics. It was he who, for the first time, demonstrated the scientifically the concepts of inheritance and variation by means of the various hybridization experiments conducted on Pisum sativum garden pea plant. Now, he was very careful in selecting the pea plant for all his experiments. But he was not just that, he was also a very passionate gardener himself. He lived in the monastery where he cultivated the pea plants and observed them very minutely. He performed various experiments on them and kept careful records of all the procedures and findings. Not just that, he even kept the data of how many individuals are produced in the offspring and how many types or of different types they are actually presented in. Then he even controlled the pollination between the plants with contrasting traits by means of features like emasculation. Later, he generalized all his results from his experiments, which were conducted throughout the span of 7 years on pea plant, that he actually generalized his results of breeding the pea plants into what we today known as the Mendel's law of inheritance. So, rightly, he is the father of genetics. Next, why did he select pea plants for his experiments? Well, the pea plant shows various ranges of contrasting or alternating traits. Like, for example, in the stem size, there were many plants of which were tall and which were also short. Of the same species, many plants were tall and many were short also. The pods in them, some were placed axially, some were placed at the terminal position. There were variations in the pod, in the form of the pod as being inflated or constricted, and the color of the pod in being green or yellow. The color of the flower also showed contrasting characters in like some plants had violet flowers and some had white flowers on them. The seed form and the cotyledon color also had contrasting variations, like the color of the cotyledon in some was green and in some was yellow, and the form of the seed in some was round and in some was wrinkled. So, what Mendel did is that he was so curious, he was so fascinated by looking at so diverse range of contrasting characters of the same species that he first developed the true breeding varieties, and then he wanted to know what would happen if I cross between the two true breeding varieties. Okay. Now, after we have seen the experimental setup of Mendel, why he chose pea plant for his experiments, we are ready to take up another question also asked from the NEET examination in the year 2017. The question is, among the following characters, which one was not considered by Mendel in his experiments on pea? The options are stem, tall or dwarf, trichomes, glandular or non-glandular, seed, green or yellow, and pod, inflated or constricted. Well, you just saw it was the option B which was not selected or considered by Mendel in his experiments on the pea plant. So, option B is the right answer. Now, as Mendel started experimenting with the pea plant, which had a diverse range of contrasting characters, after developing the true breeding varieties, what are the true breeding varieties? Means which are exactly similar to their ancestors, which are completely similar to their ancestors. So, after developing the true breeding varieties and performing a cross between the two contrasting two true breeding varieties like purple flower and white flower, contrasting characters crosses were performed, the results of the first generation of, or the offsprings which was obtained completely surprised him. What he found in his results was that only one of the parents expressed itself, and that too completely. Like, for example, if he had to cross a tall plant with a dwarf plant, he did not see any plant of the intermediate height. All he saw in the first generation was that all the plants were tall and none of the plants were short or even of the intermediate type. So that brought him to the concept of dominance. He elaborated this concept by stating that every organism has at least two alleles for every trait. Now, here I would like to tell you and explain you the terms alleles and trait. What are alleles? Alleles are the different forms of a gene. And what are genes? We know they are the units of inheritance or segments or the functional segments on the DNA which code for a particular protein. What is a trait? A trait is a form of a character. Like, for example, the height of an individual is the character, and the tallness and the dwarfness are the traits of the character or height. Okay. Now, what did he say? That every organism has at least two alleles for every trait. Now, let's take, for example, the trait is eye color brown and the eye blue eye color. The character is the eye color, and the traits are brown eye and blue eye. He said that there are two alleles for every trait. So, the brown colored eye is the dominant trait. Let that be represented by capital B. And the blue eyed trait be represented by small b. Every time it was seen that whenever capital B or the dominant brown form was present, it was always that the one form, that is the brown form, was expressed. The other form for the blue color was never expressed. It could express itself only and only when it was not present in association with capital B or the allele for brown color. So that suggested that This small b or the allele for the blue color is a recessive allele. It will be completely masked down or be hidden in the presence of the allele for brown color. So thus it is a recessive allele, and the allele for brown color, capital B, is the dominant allele. So thus he said about alleles is that alleles always have a dominant and a recessive relationship among them, and one allele always masks the effect of other allele. Now, after introducing the concept of dominance among the individuals, he performed the crosses among the true breeding types in the pea plant. Those crosses were called as the monohybrid cross. What is a monohybrid cross? A monohybrid cross is a cross between two individuals who are homozygous, who are of true breeding varieties, but only one feature or one character is taken into consideration. Right now in front of you, I have a cross between a pea plant, which is homozygous for yellow seeds, and a pea plant, which is homozygous for green seeds. It was seen that when a yellow dominant was crossed with a green, which is recessive but homozygous, all the offsprings produced were only and only yellow. No green offspring was produced, suggesting that yellow is dominant over green. Yes? Let me describe you or give you the definition of monohybrid cross. It is a cross between two types of plants of the same species when I consider or considering only the transmission of one character at a time. That is, I take into account the results of the crosses or performing of a crosses by taking only and only one feature at one time. Like, for example, in this particular slide, I have taken the height of the plant as one character, the trait of the plant, tallness and dwarfness, only this is being considered while performing the cross. In the previous slide, the trait which was taken was the seed color yellow and the seed color green. So only one feature, the color seed color, was taken into consideration here, and only one feature, that is the height of the plant, is taken into consideration here. So when I perform a cross between the tall plants and the dwarf plants, only the height of the plant is getting being considered, and the results of a cross between such a cross is that all the plants in the first generation were tall, but the genotype of those plants is capital T and small t because they have come from two parents who had the alleles capital T and small t for tallness and dwarfness respectively. Therefore, all the individuals were heterozygous in having the genotype capital T and small t. However, all of them were associated with the dominant allelic form capital T. So therefore, all the offsprings were tall. But what is the genotypic ratio of the F2 generation, or what happens after all these tall plants which I obtained in the first generation by a cross between tall and dwarf plants is performed? What is the result of the second generation? What do I get in this F2 generation? So what Mendel found was that in the second generation, he was able to get a plant having a recessive character. So when two tall plants of the F1 generation were crossed among them, he obtained a recessive plant, a dwarf plant in the F2 generation, and it had a genotypic ratio of 1:2:1. That means 3 plants out of 4 were still tall and only one was dwarf. He was completely amazed at this. Now, how come a dwarf plant be born of two tall plants of the F1 generation? So, from where has this dwarf plant come into being? He, looking into all his results from the monohybrid crosses, Mendel then proposed a hypothesis. Okay. Now, before proposing his hypothesis, his work on the monohybrid process continued with all the traits or the characters which he could see in the pea plant. Like, for example, the purple flower was crossed with a white colored flower. They were true breeding types, and all the offsprings of the first generation were all purple in color. However, when he self pollinated the plants of the F1 generation, all the purple colored plants of the F1 generation, what he saw in the next generation was that he even was able to have a recessive character, that is, a white flower plant was present from the parents, which were all purple color of the F1 generation. So Mendel counted everything that he used to obtain in his F1 generation and the F2 generation, the colors and the observations of the traits that he is studying in the monohybrid crosses, and he then applied his knowledge of mathematics and statistics to them. Then he came up with, I have already stated, a hypothesis. Now, let us go into that hypothesis. What Mendel said was that every organism has a pair of factors that control the appearance of given characteristics. Meaning to say that there are a pair of factors. Factors means the genes which we know now. What are factors? They are nothing but the genes which we know now. So Mendel said that there is something which is controlling how the organism is going to be like. And that's something he stated as were factors. Now we know they are genes. So he said that there is something, that something is factor, and that factor is coming to the offspring from its parents, and that factor is present in pairs, one from the father and one from the mother. The organism is inheriting those factors, one from each parent. Okay, and that factor is getting transmitted or transferred from one generation to the next generation as discrete, unchanging units or completely unblending units. Means they do not undergo any kind of blend in them, mix in them, or cut in them, or deletion in them, or any kind of an addition in them. It is transferred as it is to the next generation, is what was told by Mendel in his hypothesis. Then Mendel later went on to elaborate by saying that it is when the gametes are formed. Now, when are gametes formed? Gametes are formed in meiotic division, which is also called as the reduction division. So it is the reduction division which results in the formation of gametes. So during the reduction division or during the formation of gametes, what happens is that the individual, the parent, which is homozygous diploid or which is homozygous diploid, that is, has got genes in pairs coming one from each parent, it will show segregation or separation. And we know when does that happen. We have studied meiosis. We know what happens in metaphase and we know what happens in anaphase. We know that the segregation or the separation of the chromatids occur. Yes, in the meiosis, in the first phase of meiosis. So it was told by Mendel at those times, during the year 1856 onwards, he said that when the gametes are formed during the reduction divisions, these factors are separated and they are distributed as single units independently and even randomly into each gamete. Okay, those factors get distributed to all the gametes whichever are formed randomly and independently. And if an organism has got two unlike factors, for example, capital T or small t, one capital T for tallness and small t of dwarfness, then it was said that one character gets expressed completely to the total exclusion of the other character. Means, who is that one factor that gets expressed completely? The factor that gets expressed completely was called as dominant factor by him. And the factor which was not at all expressed was termed as a recessive factor by him. So, based on his hypothesis and the separation of the gametes which takes place, the distribution of the factors during the gamete formation which takes place, and on the basis of the concept of dominance, Mendel proposed three laws which are called as the laws of inheritance. Now, based on his experiments performed as monohybrid crosses, Mendel proposed the law of dominance. What did Mendel propose? He proposed two laws, law of dominance and law of segregation, based on his results on the monohybrid crosses. Monohybrid cross, and on the basis of dihybrid cross, he gave a third law which is called as the law of independent assortment. So now, since we have seen the concept of dominance and the concept of segregation of the factors, we are ready to answer another question which was in the NEET papers. I think the year for this paper was 2014. A very simple question. Take a look at it. A tall true breeding garden pea plant is crossed with a dwarf true breeding garden pea plant. When the F1 plant was selfed, the resulting genotypes were in the ratio of the following: 1 is to 2 is to 1, tall homozygous, heterozygous, and dwarf; 1 is to 2 is to 1, tall heterozygous, tall homozygous, and dwarf; 3 is to 1, tall and dwarf; 3 is to 1, dwarf and tall. So don't get confused. We have just covered the answer for this question is the first one. We have just covered that the first individual that you get in the F2 generation is has the genotype this tall homozygous. Then the genotype for tall heterozygous is capital T and small t, and the dwarf will have a genotype of small t and small t. So, after we have discussed so much about Mendel's hypothesis and the concept of dominance, I want to ask you whether this concept of dominance absolute, means, do we never see any variation in this concept of dominance as was proposed by Mendel? Well, no. There are many instances in nature where you see that there does occur variation from the Mendel's concept of dominance. The classic example is Mirabilis jalapa, or the 4 o'clock plant, which it is commonly called as. It has two varieties of flowers, red and white, of which the red flowered plant is of a dominant variety over the white. But when a cross over this dominant red was performed on the white flowered plant, ideally, according to Mendel, the plants of the F1 generation should have been all red in color. But the plants of the F1 generation, instead of being all red in color, were not red. In fact, they had a color intermediate between red and white. So, what does that show? That shows that the allele for the red color does not completely dominate over the allele for the white color. Okay, so this phenomena is a kind of deviation from the Mendel's concept of dominance, and we do not see all F1 individuals to be of the dominant parent type. Rather, it is of intermediate type. The color obtained is pink in the F1 generation, and in the F1 generation we do see deviation from Mendel's laws. However, when these pink flowered or the heterozygous plants of the F1 generation were selfed or crossed among themselves, it was seen that the F2 generation plants did hold the Mendel's ratio of 1:2:1 to be true. In the F2 generation or the second generation, the plants which were obtained were one red, two pink, and one white, which holds true as just as the Mendel's law of dominance holds true in the F1 generation. So that means to say that we do see variation or deviation from the Mendel's concept of dominance, but not in its entirety, not completely. That is why this phenomena is called as incomplete dominance. Are there any other examples of a deviation from the Mendel's concept of dominance? Well, yes. In this example, if a cross between a dominant red flower is performed with a dominant white flower, we see that the offspring that is obtained is bearing the feature of both the dominant parents. That means both the dominant parents want to express themselves and therefore get expressed in the offspring. So the offspring which results from a cross between dominant white and dominant red has features or is bearing the color both red and white in it. So this phenomena wherein both the genes of the crossing parents are dominant and both the alleles are expressing themselves is called as co-dominance, that is existing together, co-dominance and dominating together. And another examples of co-dominance we see in our human blood groups, and you can see the coat color in cattles. So this makes us ready to answer another question or take up another question, which also is our NEET question. The question is, alleles which show independent effect are called as options: supplementary alleles, codominant alleles, epistatic alleles, and complementary alleles. And you have guessed it right. The answer is certainly the option B, which is codominant alleles. It is the codominant alleles which show independent effect. Okay, we will also be studying about the concepts of supplementary, epistatic, and complementary alleles in the upcoming sessions. Hope you thoroughly enjoyed the session, and this session helped you in familiarizing you with some questions of the NEET, your examination, as well as some questions or concepts related to your topic for this particular lesson. Thank you very much and have a nice day.